Hi everybody, my name is Diego Grinman. I'm a postdoctoral associate at Yale University, and we are here on behalf of HubLE in the ASVMR 2022 to talk about science. And I'm here with Azmeer, because he's going to introduce himself to the HubLE as well.
I am Azmeer. I'm a second year PhD student at University of Rochester. I'm originally from Malaysia, but now I'm at the Wilmot Cancer Institute, and we study bone marrow microenvironment interactions with acute myeloid leukemia.
Awesome. So you want to tell us a little bit about your work here at the ASVMR?
Yeah, so I presented on one of the first days of the event, and it's really amazing. We recently published a methods paper on bone marrow microenvironment on a chip, a model to study hematopoietic stem cells and progenitor cells, and also leukemia.
Great. So, what are the key findings that you got from that work?
Gotcha. So we use a dual-channel microfluidic device, and we try to emulate the key components of the bone marrow microenvironment. These are the osteoblasts, the matrix that the cells are living in, and also the endothelium cell. And in them, we have this endothelium channel that we supply continuous flow of media during the culturing period. And what we found was that we grew these cells within our chip for 2 weeks. And one thing that you want to ask after they're growing in there is whether they're still functional. Right. So in order for us to do so, we took the cells and we performed a standard competitive transplant assay with mice to assess their functions. And what we can see is that our cells, although grown in the culture for 2 weeks, they were able to engraft and repopulate the bone marrow populations. And also they were able to give rise to multi-lineage cells within the peripheral blood.
That is fantastic. I'm very glad that it worked out. So, you want to tell us a little bit about what are the implications, for your work in the future?
Definitely. So, just looking into acute myeloid leukemia or leukemia, for example, normally people would study in 2D context. So you study stromal populations or endothelial populations with leukemic populations. But when you're doing that, you're not getting the whole sense of what's happening within the microenvironment that these cells are living in. And we've shown in our lab before that leukemic cells can interact with osteoblasts, and a lot of people here shown that as well at this conference. And these cells can change the function of the environmental cells, and then that can influence the progression of the disease. So you have to understand all of that when you're trying to look at what pathways or potential drugs that you want to study for leukemia. So for acute myeloid leukemia, right now current therapies are not effective per se for patients above 60 years old, which are the majority of the diagnosis. And the 5-year survival rate is less than 15%. So you really need a better therapy for those. So hopefully for the future, people can use our chip to study leukemia, and then we can test out drugs on the chip and see whether the cells respond to them, not only leukemic cells but also the cells in the environment. And for our lab, we're interested in looking at specific interactions within the osteoblastic and stromal cell population, because it's modular in the system, so we can knock out the receptor, for example, within the osteoblastic cell, and then assemble our chip, and then see whether leukemic cells can still interact with that particular pathway.
That is fantastic. Definitely, we need better models that reproduce the physiological concepts of the bone, and I think you are doing a great job into that.
Thanks
so
much.
What's next in your career?
So I'm very early. I just finished my second year of PhD. I'm very, very excited moving into what's next. This is my first time at ASVMR.
And I gotta say that he got the Young Investigator Award, so congratulations to that as well.
Thank you. Yeah. So right now it's just working on. We're trying to generate a human models with using human cells that we get from the clinic. So that will be very exciting, and I'm just excited to continue and do more work.
I hope you're gonna do great, and thank you so much for sharing your research with us, and looking forward to talk about more science here at the ASVMR 2022.
Awesome, thank you.